Hello, everyone. Welcome to our round two session here at Science Online Inspired, our first ever Labster conference. My name is Christopher Connor, and I am the Director of Institutional Relations for Canada here at Labster. I am responsible for developing relations with our partners in schools, colleges, and universities across all the provinces of Canada. Today in our session, we'll be discussing chemistry and biology and their approaches to leveraging technology and learning. With me today are two instructors who have spoken in many different forms about how they are utilizing technology in their courses. I would like for them to introduce themselves to everyone here. Please tell us who you are, what you do, the institution that you work for, and a fun fact about yourself that we may not know just by looking at you.
Thank you very much, Chris. Hi, everybody. My name is Dr. Felicia Vulcu. I go by Felicia, and I am an associate professor in the Department of Biochemistry and Biomedical Sciences at McMaster University in Hamilton, Ontario, Canada. Woohoo! Hopefully, you can all say hi in our chat and tell us where you're from. In the role as associate professor at McMaster, I am a teaching professor, so I mostly teach and design curriculum for lab courses, as well as drug discovery and biomedical sciences courses. One thing you may not know just by looking at me is that I'm a cat lady. That's right. I have just one cat, fur baby. His name is Norman, and he's almost 15 years old. Kate, take it away.
Thanks, Felicia. My name's Caitlin Mullarkey. I am also an assistant professor in the Biochemistry and Biomedical Science Department at McMaster University. I'm also the associate chair of undergraduate education, so I actually coordinate all of the curricula in our department, whether that's laboratory courses or non-laboratory based courses. I guess to build off of Felicia's fun fact about herself, we're not cat people. We have a dog. We have a 2.5-year-old Vizsla, who is currently my running partner. So I take him out. The beauty of working from home is we can go out for a daily run, and we've really been enjoying that.
Awesome. Great. Thank you very much. It's so nice to see you guys again. And I want to say, thank you for your time. I really appreciate the time that you've given us, as, these days you have so much on your plate. And I want to say hello to our audience. I see everybody checking in here all across the United States and Canada, and I think I saw somebody from Finland as well. So welcome, everybody. To start, as we go through our discussion on the differences and similarities in the learning approaches for both chemistry and biology, I want us to keep in mind some questions that we'll be focusing on today. Should students rely on quantitative data, or are qualitative observations just as important? Should students restrict lab reports to values obtained through experimentation, or should they provide detailed descriptions and background context? And should there be a conclusion at the end? These are just a few of the ways chemistry and biology education differ. What can two highly related fields with two different approaches learn from each other? And how can we apply these insights to leverage learning technology? Caitlin, I want to start out with you. What are your thoughts on that? What's really the first thing that you have to define when you're looking at leveraging technologies in either respective field or courses?
Yeah, great. Thanks, Chris. I think that's a really great jumping off point. So obviously the overarching theme for this session is how we marry these approaches of biology and chemistry. And being in a biochemistry department, we are well poised to answer that question, because we are integrating both of these disciplines in all of our courses, whether those are laboratory or non-laboratory based courses. And so what we're going to talk about today, or what Felicia and I would like to use to frame this discussion, is to tell you about some different ways that we are integrating different learning technologies into our curriculum, and how we can utilize different approaches depending on what our objectives are. So, just to get us started here, obviously we've already introduced ourselves, and I'm having a little trouble advancing the slides here. I don't give me a second. Okay, here we are. Okay, great. Obviously, we've already introduced ourselves. Please just call us Kate and Felicia. We're really looking forward to a very productive discussion and answering your questions as we go along. So what we really want to, we're going to frame the discussion today by talking about these 3 different questions. All right. So in the first case, how we actually use these learning technologies really depends on what we're trying to achieve, right? And going along with that, some of the questions that you highlighted, Chris, rely on our assessment methods. Okay, so traditionally, chemistry and biology have different assessment methods. Some are more qualitative, some are more quantitative. We're going to talk about how we can utilize unique and different than the traditional lab report methods to actually achieve our goals in these courses. So in the very first point, we need to decide what our objectives are in both the integrating the learning technology and also what objectives are we trying to achieve with the assessments. And we're going to take you through 2 different examples of how we've been doing this in our department, right? And then finally, we're just going to wrap up by talking about some novel technology, some really exciting ways that we've been exploring and that we're looking to use in our courses very soon. Okay, so Felicia, I'm going to just turn it over to you as we get started. So you can tell us about our second year course, our second year laboratory course, which is 2L06, and how we're using Labster in those courses.
Yes, so thank you so much, Kate. So Biochemistry 2L06 is a second year undergraduate biochemistry course that we provide in our program. All students have to take it. So let's go through it a little bit just so I can give you an idea of how this course is set up. And I'm the course instructor for this. So the way it works, it's an 8-month course offered in our department. Usually we have about 150 to 160 second year biochemistry hopefuls, as you can see here. And we've divided the course into 3 sections. So each section has about 50 to 52 students. So the way this works is I take the students through a research project, a directed research project. So instead of showing them just techniques, standalone techniques, we actually couple these techniques in a research project that spans the entire 8 months. The first term is focused on DNA. So we do a lot of DNA specific techniques. And the second term is focused on proteins. So protein work, which is what protein focus means. So every week I hang out with these students a lot. So every week we have 2 50-minute lectures and 3 hours of lab for each section. So that's right. It's quite a bit of time, but it's great to see them grow. The way that this course is divided is in 2 different areas. One is the technical skills, which includes things like introduction to basic techniques, molecular cloning, protein characterization, cell-based assays. We do focus a lot on drug discovery and things like that because our department, actually, our researchers focus a lot on infectious diseases and drug discovery, and that's kind of our theme. But interspersed within this course is the communication skills component. If you notice, it's equal to the technical skills component. So we introduce students in this second year course to oral communication, written communication, and most importantly, team-based learning. And what's really at the crux of this entire course is engaging the students in research, engaging them with each other, with the teaching assistants, with myself, with everybody in the 2L06 community. And so we do a lot of what's called flipped labs. And what that means is that instead of just going into the lab every week, we also try different engagement techniques. One of them is to leverage technology, such as Labster virtual lab simulations. And the way I leverage these in this particular course is as a pre-lab. So what that means is that I actually, in the classroom during the lecture time, go with the students, work with the students through all of the individual Labster lab simulations, and I take them through the simulation. I help them to understand what the simulation is about, while at the same time pointing out the important components of each technique and how these techniques can actually be used in a biomedical problem. And that's the really cool thing about Labster, is that they not only have the technique and the student goes in the lab to do the technique, but it's actually tied to a story. And storytelling is just about the best thing we can do to teach students, I think. We do other things assessment wise. We definitely stay away from that clunky sort of end of year or end of course report, and spread out these concepts, these objectives about scientific writing throughout the entire course. So to this end, I have lots of things called team think tanks. These are basically tutorials, small groups of students with a TA and myself, and we go through various aspects of the course together, like designing aspects of the research project, learning how to write scientific communication, presenting, so on and so forth. So the actual the other thing I always mention, because it's important, is that this will not work for you unless you create first a very safe, nurturing environment for your students to actually have dialogue. So definitely very important to set this up in your course. And I always sneak this in no matter what I present at, because it's very important to respect each other.
Yeah, that's very important. I think you're right. I know you had touched on that earlier when we had talked. I kind of want to just, I want to come back to what you were talking about a little bit later. I want to come back to what you were talking about with the, smaller quizzes and the team think tanks in a team-based experience. I think that's something really important that we should touch on. In regards to the earlier question that we had posed in the beginning about, quantitative data versus qualitative observations. What do what are your thoughts? I mean, should students rely on either one more than the other, or are they just as important? What are your thoughts on that?
That's a great question, Chris. Thank you for asking. So I can only go back to what Kate said at the beginning, because I absolutely believe in this. I believe both are extremely important, qualitative and quantitative, obviously. But what's really important is when you actually structure a course, is that you understand, you have a really good objectives of why you're doing what you're doing. It's absolutely important. So for example, if I wanted my students to just come into the research field and it's their first introduction to it, maybe I show them something like a gel or a Western blot and say, hey, here's the qualitative way of looking at this information. And then I can increase it and add a layer of complexity to it by saying, okay, now let's try and do some semi-quantitative information with this by using image J or something else where we can quantify the intensity of the bands. And from there, we can discuss the difference between quantitative and qualitative. And what's really important about that is it gives us it gives students an idea of how they can take the readout of an experiment and interpret it and outline what their assumptions are and how these assumptions can actually be controlled by the experiment. So really, it's not an idea of qualitative versus quantitative as the interplay between those two to help us understand our results.
Yeah, and I would, just to build on that, with quantitative and qualitative data, we're looking at different things, right? If you're relying on specifically quantitative data, that's more measuring a technical proficiency, okay? How well can a student follow a series of steps and get this yield at the end, or clone this particular gene. Whereas with qualitative data, that relies more on a student really engaging deeply with the concepts behind the science. And so I would say in our curriculum, we're really marrying the two, and we do that in a variety of ways based on our different assessment methods. Sometimes we have assessment methods that are more quantitatively oriented, and then we have other ones that are more qualitatively oriented. And I think it just depends on what you're actually trying to achieve with the exercise.
Exactly. That's a great point, Kate. So for example, this is just the breakdown of the 2L06 course over the entire 8 months. We usually have them divided into 2 terms. And you can see that spread throughout the actual lab time, which is the wet lab time, there's usually about 5 wet lab experiments per term. And we have 12 weeks in total. So the remaining 7 weeks are us interacting with students and helping them to develop their team-based learning skills, helping them to understand how to take the data from the labs and interpret it both qualitatively and quantitatively, and how to actually communicate this data to us through professional scientific figures, figure captions, as well as presentations to a larger audience. So all of these small assessment methods happen throughout the entire year, and they happen weekly, not only because it helps the students to get to understand the material, but it helps the students to actually process the material and gives them a more a real experience about being in a research lab. We don't go in a research lab for 3 hours a day. Like, we think about it constantly. We're constantly thinking about the experiments. And so we're trying to mimic that and stay away from the midterm final format, so that students understand that they need to learn and their learning develops as they engage more and more with the content.
It's the feedback. You want to make sure that they're getting feedback on all of the work that they're providing to you. Is that, am I reading that correctly?
Yes, absolutely. And then we can build on it. It's very important to be extremely transparent as well with why you're doing what you're doing. So I often tell my students, we're doing scientific communication today. You're going to write it in the format of a research article or a report or something. And this is how it's going to work. And then next week, it'll be, here's the same data that you analyzed. Now I want you to relay it as a presentation format. How was that going to work? What are the needs of your audience that allow you to, and how are you going to fulfill those in the redesign of that data? Because the data stays the same, it's the same result, but how are you going to communicate it?
Yes, communication. That's. I'm sorry. Go ahead, Caitlin.
Oh, no, I think Felicia makes some really excellent points there about how, Chris, some of the questions you posed in the beginning are these ideas of lab reports, right? And I think more and more we rely less on these very long, these lengthy lab reports, largely because when we have these due at the end of a project, or maybe even at the end of the term, it doesn't give us the opportunity to provide feedback. It doesn't give the students the ability to apply that and to improve their work. So in our courses, we really try to have smaller assessments throughout the term where they can get feedback, they can apply that feedback, and then actually grow in their scientific abilities from the start of the term to the end of the term.
Yeah, I mentioned earlier, I wish that had been the case when I was taking biology and chemistry. That would have been real nice to have those small little assessments instead of like this one huge project that's looming over you. So yeah, and there's some other things that you guys there's some bigger things though that you guys are replacing the lab assessment with.
Yeah, and I
think you had a really interesting view on that, on Caitlin.
Yeah,
and
so
being a little bit more practical.
Yeah, so maybe I can just shift gears. Felicia's told you about how she integrates Labster into her laboratory-based courses, and here she uses it as a pre-lab exercise. So it really gets the students thinking about how they're going to complete the labs. It gets them familiar with the order of the steps, and even in some cases, the equipment they're going to use. They can see ahead of time. Oh, well, I'm going to use a thermocycler, and that sort of thing. Now, we also use Labster in a very different way for a large service course that the department provides, which is our Biochemistry 2 EE3 course. Now, this is not a laboratory-based course. There are 600 plus students enrolled. It would be really challenging to actually have this in a lab-based environment. This course has been offered fully online since 2016. So even pre-pandemic, imagine that, we were doing courses online. And it's split between pretty evenly between some basic biochemistry and basic metabolism. So we spend about half of the course doing biochemistry and half of the course doing metabolism. And what we use Labster very differently in this course. And instead of using it as a pre-lab exercise, we're actually using it to reinforce the concepts that students learn about in the modules. So this is just the overview of the topics that we cover in the different weeks of the course. And in the first half, we're really doing fundamental biochemistry. And in the second half, we're doing very basic metabolism. And you can see that with each week, we have an associated Labster simulation. Okay, so we can't get these students into the lab, but the next best thing we can do is we can have them do a virtual lab simulation, and they become familiar with the laboratory techniques, and it helps them to reinforce the concepts that they heard about in the lecture material. So a completely different way that we're utilizing the simulations, but we found it to be equally effective. And we actually have really good feedback from the students. They like doing these simulations. They find it motivating. They find it interesting. It's a way to change up the kind of traditional delivery of curriculum.
But this goes back again to the concept of really understanding your course and what your course objectives are. So for example, for this course, our audience is non-biochemistry students who are taking biochemistry for either for their program requirements, quite a few of them for medical school pre-requirements. And so what we do with this technology is we try to show students how very basic concepts that are biochemistry in nature can actually be applied to your day-to-day, to things, biomedical problems, but also your day-to-day. So for example, weeks 6 to 10, we talk about metabolism, and we focus a lot on diet, introduction to food macromolecules, things that actually you can relate to as a non-biochemistry student. And I think that really helps them connect the content and the purpose of this content to something that's a little bit more tangible.
Excellent. Yeah, that's a really good point. And so when I wanted to ask you about, you had touched on a little bit their peer review journal that you do. And I thought it was very interesting. I was actually looking for it. I think it's called Catalyst.
Yes, right.
Yes, is that right? Yes, okay. So you have the undergrad students, okay, and then you have the grad students that review it. Is that correct?
Yeah, so it's an initiative that I started a few years ago, and it was actually started with funding science, our science fund grant at McMaster University, and I got together with some undergraduate students. It was their actual idea, not mine, and we put together an undergraduate journal that is open source and peer reviewed. It's called Catalyst: Facets of Biochemistry and Biomedical Sciences. And we've integrated it a lot in our biochemistry culture, because we're definitely telling students or asking our undergraduate students to go ahead and submit for publication in Catalyst some of their project ideas from their courses. A lot of our courses in our biochemistry program have problem-based learning concepts to them. And so these students then can submit either reviews or reports, or even multimedia, so posters or videos, and they undergo a review process by our graduate students, so mostly PhD and master students, that also get a little bit of experience in the process of peer review. So it's been a win-win for everybody.
Yeah, so you get both ends that get the opportunity to submit it, and then the other ones that get the opportunity to review it, and they both get a little bit more experience as to how science is done outside of off campus.
Exactly.
And Caitlin, what about you? What other alternatives are you looking at to the lab reports?
Yeah, so the first thing I want to say is how, just to kind of circle back to the examples
Mm-hmm.
that we've given with the different
Mm-hmm.
assessment methods. So when Felicia uses Labster as a pre-lab exercise for Biochem 2E or 2L06, it's not for marks, right? This is really an exercise where students have the opportunity to become familiar with the lab and how they're going to complete the lab. Alternatively, with 2EE3, we actually do use these simulations for marks. So
Mm-hmm.
they're not worth a lot, but they are worth a very small percentage of the student's grade each week. And to encourage them to really spend some time with the simulations and go through them more than at one time, they are for marks, but we allow the students to complete them that have multiple attempts, and then we only score their highest attempt. So that does relieve some of the pressure of answering the questions exactly right. They have some time to go through it. They have some time to go through the simulations to actually engage with the content and see the questions more than once. So in that particular scenario, again, whether you have an assessment that's for marks or isn't for marks, really again supports the objective you have for that assignment. And then, another thing I want to mention that we have been exploring in our department is using VR simulations. So the Labster actually offers 3D versions of their virtual labs, and you can access those with the Google Daydream headsets. So in 2018, we were really fortunate to be one of the first institutions to roll out this technology and have 10 Google Daydream headsets. And the difference between this is a completely immersive experience, right? Usually the Labster simulations are just 2D. You would do them on a tablet or a laptop. But with these Google Daydream headsets, you can actually turn 360 degrees and feel like you're actually in the laboratory environment. So this is a new way we're exploring of integrating novel technologies and novel assessment methods into our curriculum. We actually did a pilot study using these headsets. And just to give you a bit of background about that study, we recruited 40 students. We had them do a pre-lab quiz, so to get an idea of what their baseline level of knowledge was. Then they actually did the simulation. And then we gave them a quiz to see if they'd learned anything. And then we asked them some questions at the end about their experience and whether or not they liked these 3D simulations. The overall conclusions were that most people thought these headsets were easy to use. There were some concerns about eye strain. So the conclusion that we came to is it's important to take breaks, that you can't just have these headsets on for hours. You need to actually kind of take it off, relieve your eyes. But really reassuringly, students really had a positive experience with this headset. They thought it was motivating. They thought the simulations were interesting, that they thought that as opposed to kind of traditional teaching methods, they rated these higher. They liked them more. And they also felt like they learned something. Okay, they actually felt that they had taken away concrete skills from the experience. And so we can see that reflected in the qualitative data, but we also had ways of quantitating that because we had done this pre and post test quizzing, okay? And we can see very nicely that the scores improved when they did the simulations. So we really, this was a really interesting study to us. We're trying to figure out ways to integrate these into our different courses. The technology is admittedly the headsets are admittedly expensive. So if you have hundreds of students in your lab course, this might not be exactly the kind of magic bullet, but in certain smaller courses, there's definitely room to incorporate these. All of our evidence suggests that students like it and they learn something from it.
Yeah, I sat for hours on the couch with the VR headset and just kind of, gotten definitely lost hours of my day playing the headset, and Felicia's putting one on now, and I don't know if we're ever gonna talk to her
I'm
again.
sorry.
That might be the last we hear of Felicia. But I always tell, the people that I talk to, I say, look, using the simulations is never going to be, like Joe had mentioned at the beginning of the conference, going to be a full replacement for an in-lab experience. I don't think anybody's trying to do that. But much like, either an athlete or an airline pilot, right? Like, so if you're an athlete, you got to be in the film room, you got to get those mental reps in order to be a little bit more efficient, and you can recognize what you're seeing on the field a lot faster and you can react quicker. Or the airline pilot who, yes, please, spend as many time or as many hours as you can in the airline simulation before I get on the airplane to Hawaii. And they're going to walk away with a lot more experience, and it's going to be a lot more efficient, and they're going to be a lot more confident when they walk into the lab setting. At least that's the way that I really relate to it as well.
Yeah, definitely. And we actually have discussed, or we have some plans in the works to actually test exactly what you're saying. If students do these pre-lab exercises, does it actually help them to execute the lab? Are they more successful in completing their experiments?
I would tell you, yes. I've been a returning student. I've felt the knowledge gap. I know that, how hard it is to bridge that. And I can honestly say if I had something, similar to Labster before I went, and I'm talking about chemistry because I'm a terrible chemistry student, and it took me forever. Sorry, Felicia. I wish I had you as an instructor, but when I was a returning student, I didn't have Felicia, so I was stuck with, I definitely buried a lot of what I learned, including the instructor, whoever that was. But if I had this as a pre-lab exercise, it would have, made my experience. I would have gotten a lot more out of those courses for sure. So sorry, I didn't mean to interrupt you there, Kate. Go ahead.
No. I absolutely agree. I think that more than ever, we need to find new ways to engage students, and certainly the COVID era is a lesson for that. And yes, I would agree that in most cases, and our student data reflects this as well, they don't feel as if these simulations can replace a lab. But let's recognize that, especially in the times that we're in today, like this is a pretty good alternative if we can't actually bring people in person into the labs for a variety of reasons. This is the next best thing.
Yeah, and it's not just Labster. I mean, there's a lot of technology out there that can be leveraged to give students, a different experience, in what they're learning. Put things in a different context. And I think, generation to generation, we all learn a little bit differently. And certainly, I think, the kids that are in school today are, learn a little bit differently than certainly when, the rest of us were in school. And at least I saw that as a returning student as well. So, and Caitlin, I really want to, talk about or ask you one last question here before we get to, Q&A. You're really big on being more practical in the courses that you're teaching, familiarizing the student with, how science is done in big research studies, putting a little bit more emphasis on practical application. Is that right?
Yeah, absolutely. Our department is actually embedded in the Faculty of Health Sciences, and we are constantly looking for ways to link the basic science with the translational aspects of all of our science curriculum. So our curriculum, and Felicia is involved with a program that is part of the department, which is called the BDC program, the Biomedical Discovery and Commercialization Program. So we try and really make the content that we teach in our classes accessible to students, not just for the core scientific principles, but what it opens up to them and how it's being used in the world around them, and particularly what careers are available to them once they leave with these biochemistry degrees.
I mean, at the end of the day, we're all here because we're all in science, or most of us here, I think, are in science, and we're in science because we love it. Doesn't matter if it's categorized as biology, chemistry, biochemistry, physics, math, it doesn't matter. And so we want to show our students this absolute passion that we have for science. And the way we do that is by showing them what science can be leveraged for and what we can use it for. And one piece of that is showing them how they can grow with the science into a career that they can be happy every day. Like, I don't know about anybody else, but I'm happy every day at my job. So like, and if they can get to a point where they can identify that job or go towards that field and say, this is so amazing. They don't even know they're working. They don't even know it's a
Right.
job because they love it so much. I think that's kind of like our main objective or goal for all of our courses, or at least the ones that we teach, is to show students just the love that we have for what we have, what it takes to get there, how to be practical about it. And at the end of the day, hope that they get a career that they love. I don't know if I'm being over the top. Kate, am I being over the top? I don't know.
No, not at all. Not at all.
No. So you're speaking from the heart, Felicia.
Thank you. I'm very mindful of the time. It is 2:05. So we've basically
Yes.
gone all of our final thoughts and everything right now. I'm going to do like one last thing and say, Kate and I have a MOOC out, a massive open online course, and it's free for everybody. And if you want to check it out, come on down to the Coursera website. This is more for a general audience. So high schools, non necessarily huge science audience, but also it helps to demystify DNA. So I'm
and actually, I would also plug, Felicia, that there might be some helpful material in there for people that are teaching first-year science courses. We go through the basics of transcription and translation. So there might be parts that people could use in their courses, and it's all there. It's all online. It's all ready to go. This pre-recorded content that everyone really values these days in the online teaching era.
And it's funny. Everybody loves it.
Yes, I've heard much about it. I heard it's extremely engaging and funny, and I promise you, I'll be definitely taking a look at it. And if there's anybody wants any more information about that, I'm more than happy to provide that information as well. So I think we're going to go ahead and we're going to move over to the question and answer Q&A session here. Take some questions from the audience, and I'm waiting for them to go ahead and put up the Slido information here, so that way we can go ahead and ping you guys with a couple of questions.
I think while we're waiting, Marcos Antonio had a question. How can you transmit that enthusiasm? And it was mostly to all your students with a simulator. So, I mean, I'm a simulator, like online, transmitting enthusiasm to students online has always been, it has been definitely a challenge, I think, for everybody nowadays. I try to do things that are outside of the class time as well. So I will meet with students very often. They can ping me on, we use Microsoft Teams. I don't know if everybody's familiar with that. There's a chat feature. They get me all day long, so they can come and chat with me. I set up times to go over content with students called NIDA boosts. I set up nurture sessions if they just want to hang out because they're stressed. So I'm very mindful of mental health, their mental health. So, you know, the way I find that a lot of the times transmitting all of this community and this culture and this safety and nurture happens a lot of the times outside of the classroom, but during the classroom, you can definitely engage students in small bits. You know, take a little bit of time, cut some of the content and ask them how they're doing. Engage them with platforms like Slido, that's another technology, or Kahoot or Mentimeter, where you ask them questions and get them engaged, not just in the content. There's nothing more powerful than asking students how they're feeling. And let me tell you, they tell you the truth. So it's very important, and it's things like these that humanize the course that I think matter a lot.
They do 100%. They do. You're absolutely 100% hit the nail on the head. So let's take our first question here. What are your thoughts? And this can go to either one of you guys. What are your thoughts about using Labster solely for online lab classes at a community college? And I got really lucky. When I went back and went to a community college, it had a lot of resources, but I mean, here in the United States, unfortunately, schools, especially community colleges, are seeing less and less resources from state and local, federal levels. So, what are your thoughts about, this possibly being something that they can utilize instead of having to maintain a wet lab?
Yeah, I think again, it really depends on specifically what course you're thinking of replacing and what the students in that course are going to go on to do. So I think in certain cases, if these students are just looking to fulfill a science requirement, then it might be appropriate to do an online lab.
Right.
If these students want to go into lab careers or science-based careers, or they're going to go on to do other lab courses, there are some technical skill sets that you probably really want to introduce. So I think it depends again on, who's taking this course, what are we looking to achieve for this course, how does it fit into our greater curriculum? Because, it may be completely appropriate to do an online lab, but in other cases, you really might need to get the students in and getting their kind of like lab hands seasoned.
I agree with you. Again, my experience, there was a lot of fashion majors taking, intro to chem, and I don't understand why they have to be in a lab, because they are never, ever going to be in a lab ever again. But I guess that goes not just the instructor, but the other administrators that are making those decisions too.
If it's a big service course, I think there are a lot of pros to switching to a virtual environment. If it's a course that's going to build on other courses, or is going to be a prerequisite to other lab-based courses, I think it's a bit more challenging. So I think again, it depends on what your needs are.
Absolutely.
Yeah, agreed. Okay. So next question, maybe we'll start out with you, Felicia, on this. Did you see significant differences in using the mixed version of wet lab and simulations?
Did I see significant differences in using the mixed version of wet lab? I mean, I can only say from the data that I used in 2L06, which was self-reporting from the students. I can say the only thing I can say about that is that the students actually were able, when they came into the actual physical lab, they were able to identify the equipment much quicker, and they were able to visualize things better. So when I was teaching them in the class and we were talking about the technique, let's say polymerase chain reaction, before they would have a void, nothing to grasp so that they can actually visualize the process. Whereas after Labster, at least they could envision themselves in the Labster lab and understand how that technique works. So I think it kind of helped them to ground themselves when we were talking about, because all of the techniques we're talking about are very abstract. If you think about, I'm talking about DNA and protein work, it's abstract stuff and it's hard when you don't know the full language of it. So I would say the only thing, the thing that I've noticed about Labster is that when they were, when I was using them as a pre-lab, they were really grounded in their understanding of the practical components coming into the lab.
They had something to fall back on, they had something to visualize other than just, trying to imagine what the heck you're talking about, which is what I did the first 6 weeks of chemistry. Okay, so let's go ahead and let's go with one other question here. Okay, so can remote lecture tips on how to not get slowed down by extra student questions, over explaining to make up for not being in person and technology issues?
Yeah, so one suggestion I have that I think has been working well, and I know that every institution has different learning management systems. At McMaster, we use Avenue to Learn, which is a desire to learn by any other name. What I've been doing, because I completely understand this question, students, all of a sudden it's taking me way longer to get through my lecture because I need to feel like I have to slow down, explain things, take more questions to make sure students are actually paying attention, because you can't see their eye contact. You don't have any visual, the same visual cues that you might get in person. I have a discussion board that I post on my course page, my learning management course page. And I encourage students, if they have any questions that I don't have time for at the end of lecture, or if they have questions during the lecture, to go to the discussion board and post questions. And what I like about the discussion board is that everyone can see what I reply to. So instead of that student just emailing me and I email that one student back, everyone can see what the question was in my response to the question. Because if one person has that question, probably a lot of people are wondering the same thing. So outside of the class time, because obviously, your lecture time is limited, I keep an active discussion board that I monitor regularly, where students have the opportunity to ask questions and I can answer them.
Perfect.
I have something that Kate doesn't have. I have lab times. So I get to hang out with my students for 3-hour blocks at a time. So the way I've designed it for online this year, I'm doing online labs, equivalent of 5, I'm running equivalent of 5 lab courses, including advanced biochemistry courses, all online. And I use the platform Microsoft Teams. And so what I've done is I've created sub-channels, and I have teams of 4 to 6 students maximum. I have 72 teams. And so every week I go through and we create a lab worksheet for them, and they work with it in teams. So they're all working at the same time in teams. They've got a chat, a meeting, so they can see each other for each team there. So I can pop in, see what they're doing, and view them and talk to them verbally. And that has really helped me to really tease out on an individual level how students are how they're feeling about this information. So I use those 3 hours. I hop from team to team to team to team. It's extremely exhausting and extremely awesome to make sure that these students understand. Another little thing that I do is I will take notes throughout it. And if I see, okay, they really didn't understand how to put their primers onto the sequence, then guess what they get? They get a Felicia video. So I use another technology called Camtasia. It's a video editor and a video capture and editor. And I create very short 4 to 6 minute videos that are exactly just for that concept that I see the entire class is missing. So no bells, no whistles. No, actually there's a few bells and there's some music. But anyways, aside from that, it's just straight, like a small sort of intense moment of how to design PCR primers. And I find that works really well.
Mm-hmm.
That's impressive. That's really impressive. Thank you for sharing that. Okay, and we'll answer this last question, and I'll give this both to you guys. How does Labster allow students to address both data types? Do you require a qualitative write-up after each lab?
Yeah, so Labster, I would say, does allow students to address both data types. And for those that aren't familiar of how those simulations work, is the students are actually going through to complete tasks with an end objective, whether that's to clone a gene or to make a virus. So it does rely on their technical kind of skill set. But at the same time, what you're able to do during the simulations is to actually explore the science and the techniques and the concepts behind everything you're doing. So you have a basically, a lab notebook that if you want to, you can go and read more about the actual individual steps that are happening in a PCR reaction. Right. But then when you're in the lab simulation, you're also adding the primers and you're adding your enzyme and you're adding your DNA. So it actually allows them to do both. In terms of a qualitative write-up after each lab, I think the answer to that is, remember, we're using Labsters to kind of complement our lab courses, and we don't have lab write-ups for every single lab. And if we do have lab write-ups, they're really abbreviated. So we have kind of very short summaries of what they did and what the results were. It's not these 20-page lab reports that have 7 different sections. So I'm happy to if people want to contact us individually to ask more about the assessments, we do a number of unique assessments with the lab, including what we call bell ringers or our lab practicum. Yes. So yeah, Felicia, did you want to add anything?
Yeah, I mean, the other thing is always go back to the objective of your course. So for example, I teach the second year lab course that I specified. The main objective for this course is to bring students into our biochemistry family. And so to create that culture and to introduce them just little tidbits, just a little taste of research. So if that's the case, then I'm not going to bombard them with a written write-up after every single lab. I'm going to pick and choose what I want to emphasize or highlight, or what the takeaways I want them to have from this course. And for me, it's just an understanding of the startup of a research project, or how to at least communicate basic science. Now, when I go through my third year lab course, which is an advanced biochemistry, advanced BDC lab course, I mean, here we're talking about, quantitative PCR, the techniques, cell culture. There, I want them to really take away how to set up an experiment. I want them to be independent in designing an experiment, how to assess the readout, how to interpret the readout. That's very important. So there, my assessments would be more data analysis based. What are your assumptions? What if this goes wrong? So there, it's a very different way of thinking versus an introductory course. And then Kate mentioned another course that we do together, which is a lab course where we teach them, they actually do CRISPR-Cas9. They knock out a gene in a mouse cell line. That's really high end. These students, we've seen them in other lab courses, and there we do want them to focus a lot on the deep understanding of experimental design and interpretation.
Excellent. Thank you, guys. So that puts us right at the end of our session, but we've got a couple comments here that says, thank you for the great advice about small assessments and a lot of feedback opportunities. And then my favorite was, this is my favorite session today. Thank you. So excellent work. I want to thank you again, Felicia, Caitlin. You guys are absolutely awesome. I really, really, really appreciate your time. I know you're extremely busy. I want to thank everybody. Thank our guests again. Thank you for the events people and all the support.
Thank you,
Bye-bye.
Chris.
Thanks, everybody. Thank you.